Okay, students, today's topic is the eukaryotic cell. And what we're going to produce today is a drawing showing the organelles and their functions and how they all kind of work together. So first thing I want to say, though, is that this is a generic cell. We're trying to, this cell will contain features of both animal cells and plant cells. Okay, so just keep that in mind. So let's start by drawing a kind of a rectangular shape. We're gonna pretend that this is a plant cell. And this first line that we drew is going to be called the cell wall. The cell wall is composed of cellulose, and it provides sort of a structural support for the cell. That's what gives plants their rigidity. In fact, paper is made from cellulose, right? Wood is mostly cellulose. Okay, inside of this cell wall, we have our plasma membrane, which is composed of a phospholipid bilayer. And the phospholipid bilayer performs a much different function than the cell wall, and that is it's got something called selective permeability. And that's really important for maintaining the proper conditions inside the cell. This phospholipid bilayer is sort of the gateway for all of the different molecules that are coming in and out of the cell. And as we will learn later, the phospholipid bilayer is specialized to have some kind of control over what is coming in and what is going out of the cell. There are some properties of this bilayer that give it the ability to control the permeability. Now, I can't go into much more detail than that right now, but later we will look at the specifics for that. Next, let's go ahead and draw the nucleus. The other thing is that these items are not drawn to scale, so most of these organelles will be drawn larger than they actually are relative to this cell. So first thing we notice about the nucleus is that it's got these pores. The second feature that I want you to notice is that it's that there's two layers. So we can say that it's a double membrane. That's important. And also that it has pores. These pores are important. We'll talk about why that is in just a moment. Inside, let's say that we've got DNA in its chromatin state. That is, that it's kind of been unbound from the histones, so it's loose. It's not always in this arrangement, but we're going to depict it this way right now. So that's called chromatin, and that's a chromosome. That's a single chromosome right there. We could draw the rest. All there would be 45 others, but we can't squeeze that in our drawing here, unfortunately. All right, so let's note that connected to this nucleus is a folded structure called the endoplasmic reticulum. And it forms a continuous structure with the nucleus. It's connected. So let's label that the rough endoplasmic reticulum. The reason it's quote unquote rough is because it's studded with these ribosomes. And if you recall from freshman biology class, you learned that the ribosome is the site of transcription. I'm sorry, and that would be DNA being transcribed into RNA. So let's show that. Let's show a little piece of DNA. Oh, I'm sorry, you guys. I misspoke. This is, I had it right the first time. This indeed would be the site of translation, these ribosomes. So we're going to assume that transcription has already taken place in the chromatin. An RNA strand has been produced. And now the RNA strand is going to exit the pore of the nucleus and enter the endoplasmic reticulum, where it will then join up with a ribosome and be translated into a protein. The final product, the protein, we're going to make that a little star, will end up, after it's made, it will end up in a vesicle that will bud off of the endoplasmic reticulum and move over to another organelle called the Golgi apparatus. So this is a depiction of that vesicle fusing. So here it is. It comes over here, right? Fuses with the Golgi apparatus. And when those proteins enter the Golgi apparatus, they are modified. So we can say modification occurs here. And then once they've been modified and they're ready to be exported, a vesicle will bud off of the Golgi, and those finalized and perfected, I guess, proteins, they're packaged, modified. We'll talk more about what modifications occur later, but for now you just need to know just the basic fact that they are modified. And these proteins, of course, they could fuse with the plasma membrane and embed themselves in the plasma membrane. That's one example of what they could do. For example, insulin does that. That's an example of a protein that embeds itself in the plasma membrane. But they can do other things. They might, maybe, depending on the protein, it might go somewhere else. It might get shipped to another location in the cell. And yes, these are called vesicles. Notice too that when this vesicle up here fuses with the plasma membrane, that those proteins, they end up embedded in the plasma membrane protein. They become part of it. Okay. Yes. So we see this continuous process, right? We've got transcription occurring in the nucleus. We've got translation occurring at the ribosome, which happens to be in the endoplasmic reticulum. We then have sort of a vesicle transport over to the Golgi, where we have modification, packaging, and then sort of a exportation happening here, where the finalized protein is transported. So it's kind of like an assembly line in a sense. All right, two more organelles that we're going to draw and focus on. And let's start with the chloroplast. So again, this happens to be a plant cell. An animal cell would not contain this particular organelle. But a chloroplast is shaped much like a prokaryote, and that actually gives us a clue to its origin. It is believed that the chloroplast was once a prokaryote. We'll go over the evidences of that later, but for now, draw the chloroplast as having a double membrane. So it's got an inner and outer membrane, which means that it is a double membrane. Okay, another feature is that it's got this structure called a thylakoid. Kind of looks like a stack of coins. That's its structure. And there's a whole bunch of important redox reactions that occur in there. We'll look at the details of that in Unit 3. But for now, just know that that's inside there. And the thylakoid is dark green. It contains the chlorophyll pigment. Okay, and then there's this fluid here called the stroma. And there's a series of reactions that take place in the stroma. There's a separate set of reactions that take place in the thylakoid. We will see the details of that later in Unit 3. Okay, now common to both plant cells and animal cells is a structure called an organelle called the mitochondria. Mitochondria is similar in shape and size to the chloroplast. So it too is believed to have originated from a prokaryote. It was once a prokaryote that got engulfed by a cell and it became an organelle. Now it too has a double membrane, but it has these folds. So the inner membrane folds in. And this is the purpose of this is to increase the surface area of the inner membrane, because we will later find out that it's the inner membrane that performs an important series of reactions. And the more surface area we have, the more efficient it can do those reactions. So that's why we have this like extreme folding. The little folds themselves are called cristae. And then, so let's go ahead and label this outer membrane. Let's label this inner membrane. By the way, why do these two organelles, the chloroplast and the mitochondria, why do they both have this double membrane? The reason is because when they were taken in by this larger cell, the membrane folded in and engulfed the prokaryote, giving it an additional layer. So that's further evidence confirming this endosymbiotic theory. But anyway, looking at the structure, okay, we've got something called the matrix in the middle. So the matrix is the equivalent of the stroma in the chloroplast. It's just the fluid that's in the inside. Okay, those are the only structures you need to know right now in the mitochondria. The cristae, the matrix, the inner and outer membrane, and that's it. Okay, so I made the claim earlier that the chloroplast and the mitochondria they originated from independent prokaryotes that kind of lived as individual organisms. What's the evidence of this? And what's the theory called? The theory is called endosymbiotic theory. If we look at the etymology of the word, endo means within, sim means together, and then bio means life. So you can kind of deduce the meaning there. But anyway, endosymbiotic theory, what is the evidence of this, and what is the actual The theory is that there was once a cell that engulfed a prokaryote. So picture a prokaryote like that, right? Now, use your imagination to finish this process. If it did get engulfed, what would you have after the process is completed? You would have a prokaryote inside that has been encapsulated by the plasma membrane from the cell that folded in, right? And so that's kind of our evidence or confirmation that this is what happened, right? At least it's definitely strong evidence. So that's one of the features, right? Is that both chloroplasts and mitochondria both have a double membrane. So we can label these as evidences. So what are the other evidences? Well, if these were in fact, if they were their own organism before this took place, we would expect them to have their own DNA, some kind of nucleoid, right? And they do. They both have their own DNA inside. If they were in fact prokaryotes, we would expect them to have 60S ribosomes, and in fact, they do. And if they were prokaryotes, we would expect these organelles to be the same size and shape. And in fact, they are. Same size and shape as a prokaryote. So I don't know, pretty good evidence, right? Four separate lines of evidence. And why would this take place? Like, what's the advantage to the prokaryote? What's the advantage to the eukaryote? The advantage is that the prokaryote can perform reactions that the eukaryote can't. So obviously, there's an advantage to the eukaryote if it can team up with the prokaryote. For example, right? I mean, we haven't gone into much depth about what reactions the chloroplast can do, but it's very specialized, very complex, and it's in fact the only reaction on the planet that can take light energy and convert it into chemical energy. How about with the mitochondria? Well, the specialized reaction there is to, in a very highly efficient way, is to produce ATP from chemical energy. And we will also learn that there is a complementarity between these two reactions later. But the advantage is clear. It makes sense as to why it was in the best interest of this cell to engulf the prokaryote. What would be the advantage to the prokaryote? Well, protection, an environment where it can thrive, right? The eukaryote will provide food and resources, nutrients to that prokaryote. And so it's clear that each one has an advantage here. So this is truly a symbiotic relationship where each one benefits from the relationship. Okay, that's gonna conclude things here for our discussion of eukaryotes, and we'll go into more depth later. But these are the things you need to know right now.